Welcome to Bioscience Tutorials, where we unravel the mysteries of biology to help you understand the wonders of life. Recently, a study has been published about discovery in insulin production technology that promises to revolutionize the treatment of diabetes. In this video, we are going to explore a breakthrough discovery in insulin production, diabetes and associated problems, evolution in insulin production, transgenic organisms, transgenic cow producing insulin, and future promises in diabetic treatment. Without any further delay, let's get into. Recently, a study was published in the magazine Cosmos about a team of Brazilian scientists who have produced a cow which can make insulin in its milk. We know that insulin is mainly used to treat diabetes by keeping blood sugar level under control. According to recent reports of World Health Organization, about 422 million, that is 42.2 crore people are diabetic, and about 1.5 million, that is 15 lakh, dies because of it annually. As per the article published in a popular newspaper, The Hindu, 2021, India is a house to 101 million people with diabetes and 136 million are pre-diabetic. There are two types of diabetes, type 1 and type 2. Type 1 diabetes occurs when the body stops producing its own insulin. And type 2 occurs when body doesn't make enough insulin and become resistant to it. Diabetes is a chronic metabolic disorder characterized by an elevated blood glucose level, which leads over time to serious damage to heart, eyes, kidney, blood vessels, and nerves. Insulin from cattle and pigs was used for many years to treat diabetes and saved millions of people, but it wasn't perfect as it caused allergic conditions in patients. The first genetically engineered synthetic human insulin was produced in 1987 using E. coli bacteria. Still, there were certain challenges involved with the production of insulin using recombinant DNA technology, as getting insulin assembled into its mature form. The process involves formation of a primitive form of insulin called proinsulin that contain C-peptide, which is not a part of mature insulin. Again, for decades, insulin manufacturers have claimed that high market price of insulin are necessary to spur innovations and investment in research and development. Nowadays, commercial insulin like Humulin, Actrapid, and Insuman is mainly produced using bacteria like E. coli and yeast Saccharomyces with the help of recombinant DNA technology. In this technique, the targeted organism is introduced with a piece of DNA, that a gene responsible for a synthesis of desired protein from another species like humans. Their modified organisms are known as transgenic organism. This time, a transgenic cow with a human gene has been produced using the same technology. This research is led by Matt Wheeler at Department of Animal Science at University of Illinois, United States, who says Mother Nature has designed the mammary gland to make the protein really efficiently. We can take advantage of the system to produce a protein or hormone that can help hundreds of millions of people worldwide. A team of scientists at University of Sao Paulo, Brazil, inserted a human gene coding for protein insulin into the nuclei of embryos of about 10 cows. They were implanted in the uterus of the normal cow which resulted in the birth of one transgenic cow. Attempts were made at impregnating the cow once she had reached the maturity were unsuccessful, so the team stimulated lactation using hormones. Basically, their goal was to make a pro-insulin, purify it to insulin and make available in usable form. But the good thing here is that herself she makes about 3 to 1 biologically active insulin to proinsulin. The insulin and proinsulin were expressed at a few grams per liter of milk because the milk volume was smaller than expected due to the hormone induced lactation. The researchers can't say exactly how much insulin would be made in a typical lactation. Scientist Matt Wheeler says a typical unit of insulin equals to 0.0347 milligrams, and consequently, if a cow makes 1 gram of insulin per liter, that means each gram is equivalent to 28,818 units of insulin. A Holstein cow can produce 50 liters of milk per day. Now you can imagine the quantity of milk being produced. The insulin is need to be extracted from the milk and purified before its use in humans. The insulin producing cow is a proof of concept that could one day be scaled up to produce enough insulin to supply the diabetics worldwide. Scientists are now planning to reclone the cow and attempt pregnancy and lactation in the next generations. Eventually, a transgenic bull will be produced to mate with the female cow, creating transgenic offsprings that can be used to establish a purpose-built herd. In conclusion, the recent discovery in insulin production technology marks a significant milestone in the quest to improve diabetic treatment and care. Stay tuned for more valuable updates. Don't forget to like, share, and subscribe for more educational content. Until then, stay curious and keep exploring.